Assalamualaikum warahmatullahi wabarakatuh and good afternoon to the fellow audience. We are STP students from Group 10. We'll be presenting about our project titled Green Adsorbent for Water Filtration System. Our group consists of 4 members, me, Nur Izzah, Adam, Madeline Teo and Melanie Rachel, and has been supervised by AP Dr. Cecilia and Mr. Faisal. First of all, let me briefly explain about the introduction of this project. For the background, we have identified that the presence of heavy metals in rivers, particularly the lead aka plumbum and copper, severely affects the water quality and also imposes detrimental effects in human health. In order to remove the heavy metals, iron liquid is very effective and has been widely studied since iron liquid can be recovered by stripping, then it also can be reused several times for liquid extraction. Iron liquid is also considered as eco-friendly, which is a green adsorbent. It has very unique properties such as no vapor emission, good solvation ability, higher thermal stability, and tunability. While for the problem statement, it is identified that the current method, which is only applying iron liquid in liquid form as the adsorbent for heavy metal extractions, has some drawbacks. The few drawbacks such as we have to use large quantities of iron liquids and there is also possible loss of iron liquid into the aqueous phase due to the incomplete phase separation. So because of these issues, there is an increased demand to find alternative technologies to overcome the drawbacks of heavy metal extractions. So an answer to these problems is we impregnating the iron liquid into the activated carbon as the solid supported iron liquid, and that could be an alternative materials as a green substitute, and also it could enhance the removal process of the metal ions. Next, there are 3 objectives that we are implementing in this project. First, to produce an iron liquid that is able to remove heavy metals. Second, to produce iron liquid that can be incorporated with our activated carbon as the water filter adsorbent. And third, to determine the metal removal effectiveness of a water filtration system with green features in the water filter adsorbent, which is the recyclability.
For our literature review, we found that activated carbon is an effective adsorbent owing to its well-developed internal microporosity and broad range of surface functional groups. Activated carbon is also easily obtained from high carbon content trash in the environment. Also, due to its larger surface area and chemical complexity of its surface area, activated carbon has a high potential for absorbing heavy metals. Activated carbon enhanced with ionic liquid is also shown to increase heavy metal absorption efficiency, as proven from the absorption of imidazolium-based ionic liquids onto activated carbon. Besides, increasing length of arcuate chain increases hydrophobicity of the ionic liquid, which further increases its absorption capacity. Therefore, ionic liquids with longer structures will have higher capacity for absorption on activated carbon.
I will share our design concept. So we have drawn a design for our water filtration system where we have combined the chemical and sediment filters into one filtration system. So the reason why we choose these 2 filters is to increase the effectiveness of the filtration. The chemicals filters used for this project was activated carbon, where it is responsible to remove any dissolved chemicals in the sample that will pass through the system later. While the sediment filters was responsible to filter out any visible impurities like maybe dust or debris. So for the arrangement of the layers, which is the filtration unit, we have been arranging the layers of sediment which are very coarse sand, medium coarse sand, and also fine sand by referring to the Wentworth scale. So the layers are arranged based on the size, where the larger size will be on the top and the smaller size will be on the bottom. So the activated carbon was put in the middle of medium coarse sand and also fine sand because the size of the activated carbon was in between the size of the medium coarse sand and also the fine sand. And lastly, we end our layers, the filtration units, with a layers of cotton in order to prevent the leakage of the sediment and activated carbon into the filtered sample later. Next, we have also prepared the sediment filter by sieving about 2 kg of sand using the sieving machine in order to separate the sand into various grain size. The sand of various grain size collected was very coarse sand, medium coarse sand, and also fine sands with the range size of 1.18 mm to 115 micrometer. So the next material that we prepared was the activated carbon. So we have been using about 200 grams of activated carbon. So the activated carbon was first heated up to 110 degrees Celsius in a vacuum oven in order to remove water traces from it. It is then put in a desiccator to prevent any moist accumulation in the activated carbon during the cooling method.
Okay, so to prepare the ionic liquid, we need to synthesize 1-butyl-3-methylimidazolium chloride, which is the BMIMCl, and sodium dicinamide, which is the NaDCN, to produce 1-butyl-3-methylimidazolium dicinamide by using metathesis reaction. We will obtain the sodium chloride, which is the salt, as the byproduct. Based on the stoichiometric calculation, 1 mole of BMIMCl will react with 1 mole of NaDCN to produce 1 mole of BMIMDCN. So we need 50 grams of the product to be used in this experiment. So we need to calculate the mole of BMIMDCN, which is by dividing mass with the molar mass. So we'll get 0.0731 mole. As 1 mole of both reactants, we will produce 1 mole of the product. So the mole of both reactants will be the same, which is 0.0731 mole. So we need to calculate the mass of both reactants. So for BMMCl, we need to multiply the mole with the molar mass. So we will get 12.77 gram. While for NaDCN, we will get 6.509 gram. So after calculating both reactants, we need to mix both of them to be dissolved in 50 ml of acetone. Then it will be stirred for 20 hours using a magnetic stirrer at room temperature. After that, it will be sealed in round bottle flask using paraffin film, and it will be kept at negative 20 degrees Celsius for 5 days. Then the mixture will be filtered using filter paper, and it will be evaporated using a rotary evaporator. It will produce a colorless ionic liquid instead of slight yellowish. This is because we did not perform the purification using column chromatography. After that, the product is sampled and we determine the structure using NMR and FTIR analysis.
After we have synthesized our ionic liquid, we sample the ionic liquid for NMR analysis in order to determine the structure of organic compounds in the ionic liquid. The NMR spectra was recorded on Bruker at 1200 series spectrometer at 500 MHz with deuterated acetone as our deuterated solvent. Besides NMR, we have also sent a sample of the ionic liquid for FTIR analysis to verify the anion component. The FTIR analysis was performed on protein Elmer Frontal 01 model spectrometer.
The impregnation of ionic liquid into the activated carbon is prepared by the incipient impregnation method at room temperature. The materials were prepared with 4 different loadings of ionic liquid onto the activated carbon. First is 0%, second is 10%, third is 5%, and fourth is 2.5%. And the mixture is gently mixed with methanol and stirred for 15 minutes at room temperature. Methanol is then slowly removed using the rotary evaporator that will be resulting in physical immobilization of ionic liquid into the activated. And finally, the impregnated of activated carbon is dried and stored into the desiccator. And the filtration process is conducted at the water filtration system at lab 200105. And then the contaminated river will flow through the layers at the top of the filtration column. And then the product will flow out from the bottom of the filtration column.
Lastly, we have also performed AAS analysis for our project. AAS determines elements in the sample, which in our case is to measure the concentration of lead and copper ions in our samples to test the efficiency of the produced absorbents. The AAS analysis was performed on Agilent 240 Fs, and using the concentrations of elements obtained from AAS, we'll be able to calculate the efficiency of the absorbents using the formula shown.
After we obtain the group, which consists of me, Izzah, Madeline, and Rachel, and we got already our topic, we do discussion among each other and we do literature review as suggested by our supervisors, which is Dr. Cecilia and Sufaisal. Then we do task distribution with each other, with Izzah being the group leader. Then we prepare the activated carbon, which is done by Izzah, and ionic liquid, which is done by me and Madeline. After that, we analyze the ionic liquid using NMR and FTIR, which is done by Madeline and Rachel respectively. After that, we incorporate the ionic liquid into activated carbon, which is done by Izzah. And then the sediment filter system is prepared by Rachel. And then we collect the river water source from Kinta River. And then all of us do the filtration process at block 20 and we produce 6 types of sample. And then all of us we do analysis using AS based on the 6 samples that we have done.
Starting from the NMR analysis, a total of 15 hydrogen atoms have been detected despite slight traces of impurities found in the ionic liquid, possibly due to the lack of the purification process after synthesis. The peaks are then assigned to the different structures in the compound as per the multiplet report generated for the analysis, and with final confirmation, all 15 hydrogen atoms belong to the beaming cation of our ionic liquid.
Moving on to the next part, for the FTIR analysis, we have used this analysis to get a confirmation on the structure that we have in our ionic liquid. So basically, we have detected about 6 peaks that can be used to analyze the structure of the ionic liquid that we have. So the peak is, and the peak that we found in the graph is they refer to the IR correlation table in order to identify the type of bond axis in certain frequency range. As you can see here, all of the stretch found in the FTIR graph can also be found in the expected structure in the ionic liquid, starting from the NH stretch to the CN stretch.
So this is the result we obtained from analysis of AS. So the sample names are labeled below the slide. So to conclude, all the samples concentration after filtration, which is number 2, 3, 4, 5, 6, decreases compared to before filtration, which is the number 1. Filtration using the 10% by MDCN, which is the number 5, produced the highest efficiency to remove the CU by 11%. The recyclability, which is the number 6, is proven work as actually managed to reduce the amount of CU compared to before filtration. However, the results are affected by errors during the experiment. For example, like contamination in the activated carbon and leakage during the filtration. As the previous slide analyzed CU, this is the slide analyzing the SPB. As we can see, all filtered samples concentration increases compared to before filtration. This is not logical as it does not follow the literature at all. So we made some inference. Maybe the river water contains very little amount of lambum. The river water that we had does not store properly, and it is difficult for us to determine the change of concentration from very tiny to concentration. So we conclude that the results are invalid. So to overcome the previous problem that we occurred during the experiment, we rerun another analysis, but instead of using river water, we use 1 ppm of plumbum. So we run filtration using blank 2.5% at 5%. So for 2.5%, it is higher than blank because of errors during the experiment. So after we overcome the errors, we managed to get the correct value for 5% as it produced efficiency of 55% that can filter out the plumbum. However, we cannot obtain any result for the 10% as we did not have enough sufficient amount of product to be run the experiment. So to conclude, our filtration system actually can purify CU and plumbum.
So for this project, we have spent about 195 ringgit and 51 cents, which is for the preparation of our ionic liquid, which are BBMCl, and also sodium diacetylamide, and also our activated carbon, which is the charcoal powder. So we have spent about 123 ringgit and 33 cents for our BBMCl, 47 ringgit and 48 cents for the sodium diacetylamide, and also 24 ringgit and 40 cents for the activated carbon. So from the quantity of grain absorbent that we have successfully compared the efficiency of heavy metal removal for lead and copper by 4 different loadings of ionic liquid into activated carbon. So the highest result shown, 20% of lead and about 11% of copper can be removed from the initial sample.
As a closure, the higher the concentration of ionic liquid in activated carbon, the higher the efficiency of heavy metal removal from water during filtration, as proven by the results for 10% diamine diacetylamide incorporated activated carbon solvent, with the highest efficiency in copper metal extraction. We expect a similar outcome for lead removal efficiency from 10% bimidazimide incorporated active carbonate solvent, as deduced from the AAS result for the filtration performed on 1 ppm lead standards using 5% bimidazimide incorporated active carbonate solvent. Ionic liquid enhanced active carbonate solvent also retains moderate efficiency in metal extraction, as observed from 10% bimidazimide incorporated active carbonate solvent after HCl rings. It is expected that a similar outcome for the solvent will be obtained for lead metal extraction. Overall, the objectives of this science team project has been achieved.
Since we only obtain very low percent of heavy metal removal, we could actually take into consideration of some recommendation to increase the removal efficiency. First, increase the hydrophobicity of the ionic liquid. It could favor the transfer of metal in the ionic liquid phase via the cation exchange process. Second is removal of water and impurity before impregnation, and structure analysis could also be taken into account to make sure a perfect impregnation and could produce better structure analysis. And last but not least, we could also study on the effect of pH for the extraction capability, so by that we could compare for better heavy metal extraction. Finally, these are some of the references that we have referred to in this presentation. This marks the end of our presentation. Thank you. We may now proceed with the Q&A session.